Hi Year 12s, welcome to the first PowerPoint and video for our next topic. Our next topic is going to be looking at homeostasis in general. We're also going to be looking at this PowerPoint specifically at tolerance limits and tolerance ranges. So let's begin. As per usual, here's your need to know. This is quite a small need to know for this topic. Overall, the entire topic of homeostasis has a very small need to know. So if, again, as per usual, if you want to take these down, just remember to pause the video here and write them down, but we're not going to go through these specifically. This is just what's being covered in the video. So, first off, tolerance limits. What is a tolerance limit? Well, to understand that, you have to understand that every species of animal or organism on Earth has a range of tolerances for different abiotic factors. Okay? And those organisms will perform more successfully and more effectively when they are living in their optimum tolerance ranges. When I use the word tolerance range, what I'm referring to there is the difference between the minimum and maximum levels of which an organism can survive and work efficiently. Organisms that have a really wide range of tolerance ranges, so they can live almost anywhere, are obviously going to be found more on Earth than those that have narrow tolerance levels that need very specific conditions. Because those ones with narrow tolerance levels have to pick an environment that caters specifically to their needs, and they'll only survive in those areas. So the range of tolerance limits that you have determines how widely spread you can be across the Earth. There are a number of different tolerances that we need to talk about, the different abiotic factors. We're going to start with body temperatures. So, body temperature refers to an organism's tolerance range to temperature. So, what temperatures can that organism survive in? For humans, obviously, we optimum around 37 degrees. Now, organisms, to help with this, they'll regulate their body temperature. So that means that they can control their temperature to help their metabolic reactions occur at the rate that supports their life processes. If those temperature falls below the tolerance limit, enzymes are going to slow down, which means they're not working efficiently, which means the metabolic reaction rate is too slow and life is not maintained. On the other hand, if the temperature rises too high, then the tolerance limit is again passed and the enzymes may start to denature, which means the enzymes aren't going to work at all, which means the rate of metabolic reactions decreases or stops completely, and therefore, again, life doesn't continue. Controlling body temperature with narrow tolerance limits actually allows organisms to not be affected by changes in the environmental temperatures. So for us, we have very narrow tolerance limits for temperature. 1 or 2 degrees above 37 degrees for too long can actually sustain brain damage and cause lots of problems. So having our body with that very narrow tolerance limit enables us to be unaffected by changes in the environmental temperature. We and other animals use thermoregulation to control our body temperature within that narrow tolerance range. Water availability. So water is really important for lots of our life processes. So we need water for transport. So lots of molecules actually will dissolve in water in order to be transported around the body. Our metabolism. All of our enzyme catalyzed reactions actually occur in solution. Most of the time that will be in a water solution. Thermoregulation. So water has a very high specific heat capacity, and therefore that's one of the key components that helps us maintain a constant body temperature. Movement. Our muscles use water a lot in muscle movement. Sexual reproduction. A lot of animals that will sexually reproduce will use water to bring the male and female gametes together. And lastly, just for support. So water-filled cells provide structural support for different tissues in animals and plants. Think of the vacuole in plants. So water is really important, and you have to make sure it's keeping within the right balance in the body, the right tolerance ranges and the right tolerance limits. So when water does fall below those tolerance limits of an organism, their cells and tissues will shrink and become damaged. That can also lead to more solutes being more concentrated, such as sodium, which will affect the folding of polypeptides and proteins, which in the long term is going to inhibit the activity of the enzyme. Alternatively, when the water levels rise above the tolerance limits, tissues and cells will start to swell and burst. That's going to cause some solutes to become too dilute, which is going to reduce the rate of life processes again. Osmolarity, another really important one, talking about the concentrations of solutes and solvents in the body. What osmolarity actually is just the concentration of a solution expressed in units of solute particles per litre. And you've got your units there on the PowerPoint that you might like to note down. We've also got microorganisms, plants and animals, all needing to worry about osmolarity, and they all control it via a process called osmoregulation. Now there's two types of osmoregulation. There's osmoregulators and there's osmoconformers. Osmoconformers have the exact same osmolarity as their environment, and whatever the osmolarity of the environment they live in is, that's what their body will also be like. So think about fresh and saltwater fish. Whereas osmoregulators, like us, we closely regulate our internal osmolarity to prevent it from changing as the environment's osmolarity changes. Blood glucose level, another really important one. Why? Well, because glucose is the primary sugar that we use for respiration. It's controlled in order to ensure that all of our cells receive the right amount of glucose to ensure that the required levels of cellular respiration actually occur. The normal concentration of glucose in the blood for humans is around 3.9 to 5.5 millimoles per litre, which is mmol/L. We call that the fasting level because that's the level it should be after you've fasted for a set amount of time and had your blood taken. Consequently, this is also the tolerance level for glucose in humans, so it's what we expect humans' blood glucose level to be at. If glucose levels in the blood fall below the tolerance limit, then the rate of respiration becomes too low because you don't have enough glucose, and life processes aren't maintained. What can happen is a situation called hypoglycemia, and remembering hypo sounds like low, so glucose is too low, hypoglycemia. If hypoglycemia is left untreated, it can lead to a loss of consciousness and potentially the patient entering a coma. On the flip side there, if glucose levels in the blood are too high, we get something called hyperglycemia. So whenever something's low, it's hypo. When it's high, it's hyper. So hyperglycemia. What happens here is it causes more water to diffuse into your urine, and the patient urinates more frequently. Because that's all because aerobic respiration is occurring at a faster rate because there's more glucose, which means the patient is also going to eat excessively to replace the glucose that they're losing, and you're constantly getting high rates of aerobic respiration, and you end up having that more water which goes into the urine and is excreted. Again, like hypoglycemia, if hyperglycemia is left untreated, it can cause coma and death. Carbon dioxide concentration. So carbon dioxide, as we know, is a product of our aerobic respiration. We consider it as a waste component of that. Generally, it's kept around 5 to 6% in the human blood. At those levels, it's considered a non-toxic gas, and it helps to regulate and control the rate of depth of breathing. So how much carbon dioxide you have in your blood determines the rate of breathing for you. If your carbon dioxide concentration falls below the tolerance limit, you get something called hypocapnia. Now again, there's that word hypo for low. Hypocapnia generally results in respiratory alkalosis, due to the decrease in hydrogen ions in the blood. So basically your blood becomes too alkaline. On the other hand, if carbon dioxide concentration rises above that tolerance limit, you get a condition called hypercapnia, which again is due to carbon dioxide levels being high in the blood. This leads to a process called acidosis, which means your blood is too acidic. The different processes that we look at for our tolerance ranges are thermoregulation, osmoregulation, glucoregulation, and chemoregulation. We've talked about some of these already, but thermoregulation, as the name suggests, is the regulation of temperature in the body. Thermoregulation. Osmoregulation is the regulation of the water and solute concentrations in your body. So osmo. Glucoregulation is the regulation of the blood glucose level in the body. So hence the gluco in glucoregulation. And lastly, the controlling the blood pH, which is actually your carbon dioxide levels in the blood, is done through chemoregulation. And that's all controlled by the breathing rate. lastly, just a brief look into nervous system and homeostasis in general. So we need to look at the stimulus response model, which is truthfully something you've been looking at since around year 9. And knowing that it starts with a stimulus. When your body senses that stimulus in the sensory receptor, it sends a message to your control center, in this case our brain or central nervous system, which will send a message to an effector, which will initiate a response. A sensory receptor is the organ in your body that responds to a stimulus, and it ensures that sensory information is being transmitted via nerve impulses to the control center of the nervous system. The control center is responsible for receiving and processing that information, and then sending a signal to the effectors via either nerve impulses or hormones. And the effector, lastly, will respond to nerve impulses or hormones by opposing or enhancing the stimulus depending on positive or negative feedback, which is what we're going to talk about next. So for humans, for example, for us to survive, we need constant temperature, constant pH, constant oxygen concentrations, and we need osmotic balance. We also need to remove the waste from our body, such as carbon dioxide and urea, because if they build up, they can impair our normal cell function. Now we do this through control mechanisms. There are 2 types of stimulus response model: positive feedback, negative feedback. In positive feedback, so let's say carbon dioxide concentration in the blood, the stimulus is that there's too much carbon dioxide in your blood and you're going through acidosis. So that's the stimulus. That sends a message to your control center. In positive feedback, the control center would say, okay, to fix this, we're going to decrease the breathing rate, meaning we're building up more carbon dioxide in the body. So it would cause the human body to spiral out of control and the blood would just become more acidic. So positive feedback doesn't work, because positive feedback is the body responding by enhancing the stimulus. Negative feedback, on the other hand, is what our body actually goes through. So same procedure. You've got too much carbon dioxide in your blood. You're going through acidosis. Your control sensor finds this out through the sensory receptors. Its response is to increase the breathing rate and extract the carbon dioxide from your body, and therefore inhibit the stimuli that was actually there to start with. It results in control and inhibiting the stimulus to return your body to those tolerance ranges that you need. That whole process of maintaining a stable environment we call homeostasis. The mechanisms that achieve homeostasis we refer to as homeostatic control mechanisms. What are homeostatic control mechanisms? Well, they're self-regulating mechanisms that involve negative feedback. Think of it like a gas hot water system. So when the water is too cold, the gas turns on and a flame comes on to heat the water up to a certain temperature. Once it reaches that temperature, say 28 degrees, the flame turns off. The water starts to cool again because there's no flame, and the flame turns back on and repeats the entire process. The whole time the water is fluctuating around that 28 degrees Celsius. That's the same way that homeostatic control mechanisms and negative feedback in your body work.